also coordinating a European project called RAIDS, Rational Assessment and Innovative Drug Selection, which is on cervical cancer and which is just a clinical study biopsying patients before and after standard treatment and following them up with liquid biopsies up to 18 months. So there is many different alleys in immunotherapy, and it's a bit confusing for the clinician, and I try to help you through the meanders of this immunotherapy world. The main question is, how can we successfully activate the immune response in cancer, and how can we track immune responsive cells who just pass in the circulation fleetingly? We often do not have access to the tumor microenvironment, which I think is a very important variable to study. We also do not have access, as the classical immunologists do, to tumor-draining lymph nodes very easily. It can be arranged in some circumstances, but it's a difficult thing to achieve. Now, I would like to give you a home-take message in a nutshell. If we compare immunotherapy to controlled flight, in controlled flight we need propulsion, which so far has been essentially adding antigens and adjuvants to patients. We need lift and wings, which I would compare to the tumor microenvironment and its disturbances. And we need a balanced center of gravity and weight, which I would like to compare to checkpoint inhibition or modification. Now, the biological prerequisites for immune function in the context of cancer have been very well elucidated by classical immunologists in many laboratories in the world. And most people come up with 4 steps, and I would like to actually make it into 3 steps. So there's 1 antigen presentation, still we need that. There is dendritic cell maturation and T cell expansion, which I would put together into the microenvironment, the wings section. And there's then prolonged CTL activity, which can be achieved by checkpoint modulation. I would also like to put your attention to the T regulatory cells and the myeloid-derived suppressor cells, which will, whatever we do, even if we take the dendritic cells out and mature them in vitro, and if we take the T cells from the patients who have reacted a little bit out and expand them in vitro and put them back into the patient, though they will still be faced with this microenvironment, which is not favorable for an immune response to be prolonged. So again, back to the aviation model, crosswinds. We can take tumor-infiltrating lymphocytes out of the crosswinds, and we can expand them in the laboratory in a controlled and defined environment. We can do the same for dendritic cells, and this has already led to reagent to be authorized in the US in prostate cancer. Now, today we are going to hear more about T-cell expansion and also about not only complete T-cells, but just taking the T-cell receptor of a reactive T-cell out and putting into a little vector and bringing it back in a defined fashion into patients. And we learn more about prolonged T-cell activity as well about rational combinations. So in order to treat the microenvironment, we can decrease myeloid suppressor activity. My personal interest is in tumor-associated macrophages, and I think that's something we should really look at. We learned yesterday that with a little bit of radiotherapy, we may switch them around from M2 to M1 macrophages. There are many reagents also now coming into the clinic which can block the CSF1 receptor interaction. Then we oncologists have many standard therapies under our sleeve, and we should use them accordingly. If they are active, we should use them together with immunotherapeutic modulators, keeping in mind that we should really assess patients' microenvironment, maybe draining lymphocytes, draining lymph nodes, before and after any reaction, so that we get a better feeling about how we could adapt it. And I think each patient needs to be considered separately. Each patient has his own microenvironment, and we need to adapt to that. And then finally, the modified checkpoints, and we learned on Saturday that there's preclinical data that if you combine PD-L1, PD-1, and CTLA-4 inhibitors, we may be much more active than using one alone, and there's very good scientific reason to do that. Now, about toxicity, we don't really know about these things yet, and so we have to always carefully watch for toxicity that may appear, which could be autoimmunity, and there may be always some autoimmunity associated with an immune reactivity to the tumor. We just have to make sure it doesn't get out of hand. And then I would like to leave you again with that message that if we grow monocytes in culture outside of the tumor microenvironment in GM-CSF and IL-4, and we add then at the last step for 24 hours MCSF or CSF1, we end up with a completely different cell than if we did do that same thing with LPS. We see that these cells are phenotypically and functionally different. Now, this is what happens in the patient, and this is what we would like to happen. Just on checkpoint inhibition, this is a waiting model for Horsa glider back to aviation. These were gliders, quite fragile structures that were used in the Second World War for the Allied landing in Normandy just before D-Day, and to make sure that they had a perfect center of balance, every person was weighted and every material was weighted and put at a precise spot into the plane, so that the general center for gravity was situated somewhere here. And if the plane is centered too far backwards, you can imagine it will not take off unless you power it with a rocket, or if it's centered too far forwards, then it tipples over. And this might be metaphors for cytokine storms or for autoimmunity. And these are shown here. So what we do now with checkpoint inhibition is we are most interested in those here, but there are others already that are evaluated in the laboratory. And I would like to say one word of caution. There has been an accident, and this was in the New England Journal of Medicine in 2006, when healthy volunteers were given an agonist for this one to stimulate their immune system, and this ended up with 6 healthy people being in intensive care, and 2 of them for several weeks. So there can be dramatic consequences, and so to go into innovative checkpoint inhibition, I think it shouldn't be done in a private clinic, but it should be done in a center where you have anesthetists and people who can resuscitate if something did go wrong. Keeping in mind so that in the cancer patient, the immune setting is probably more conservative and not that easily activable. So I would like to leave you with this image now of the future for immunotherapy is ready to take off. And the presentations today will deal with tumor-infiltrating lymphocytes, and that's my co-chair who is going to speak about that. Then we hear about adoptive therapy with engineered T cells from Carl Jun, and new development in clinical vaccination by Ulrich Keilholz, and then rational combinations by Dr. Mellerio. And my chairman, who has not appeared yet, will speak about clinical perspectives.